And so I decided instead I would present what I learned and what worked for me in understanding things, because there's a lot of background that people present with statistics that isn't necessary unless you're a statistician, you know. Instead of, it gets in the way of you learning the basic tools and understanding what you're trying to do and what you're trying to accomplish and how you're trying to work with the data. And so that's what I'm trying to present here, is just the skeleton. You know, if you want to get more in depth, you know, the formula for standard deviation, you can look that up. You can always look that stuff up. And as you use it more and more, you'll learn it and you'll memorize it. Same as everything with variance, standard error, again, you'll learn those things with time. You know, there's no point in having to include that here because you can Wikipedia it. You guys are millennials. You guys know how that works, okay? So, first up with stats, when you start a study, it's going to fall into one of two categories. You're either going to be doing just a descriptive study, where you go and you find something and you want to look at it. Like, okay, what is here? So like a lot of times when you're doing diversity studies, or you're looking at richness or evenness or abundance, they're just descriptive, because you're just trying to see what's here. Okay, whereas when you're doing hypothesis testing, you go out there with an objective. There's a question you have that you want to answer, and it's not what is here, instead it is what is happening. And so what you're doing is you're trying to see and draw conclusions based on something. So, for example, my group was trying to see if plant biomass changed as a function of a nutrient concentration. So that's kind of thing, you're framing it. So they hypothesized that as you increase nitrate, you'll have more plant biomass. That is an example. Whereas if they were just going out and like, we want to know what plants are here, that's a descriptive study. So you want to frame it and think, okay, what is my study? Which one does it fall into? Because from there you can then start going and asking and realizing what statistics you're going to start using. So the reason we really use statistics is because when you enter all your data, it looks something like this. This is, you know, just, it's another example of stuff, but it's a lot of business and money and whatever. But this is usually what you end up with, and this is only 34 lines. Heather Cramp, whoever is in the fish group, she's working with a data set that's about several 100,000 lines of data. And so it makes no sense to try to sit here and like click buttons and be like, I want to know the mean for these two, you know, or something, because it's just so vast. And so that's where statistics comes in. It helps simplify that and make it more, make it simpler for you to understand what's going on, because here you can't tell anything. You don't know what's happening. And the reason that this is a problem is just it's extensive, it's unorganized, and it's indeterminate. You can't determine anything from the raw data just looking at it like that. And again, extensive. It could be up to hundreds of thousands of lines. And so that's when computer programming becomes especially good. So definitely look into doing that when you guys are older. It's one of those skills that now in our technological era is invaluable, and you really, if you have a good job, if you do a good job of coding and you learn it, it'll help you get jobs for sure. Okay? So to make sense of data, there's two types. All right. A lot of times people use, you know, well, no, that was another thing I was going to get to later. So you have either continuous data or categorical data. All right, and they're different. So continuous data, you guys talked about the other day. I remember hearing it when we were doing the graph out by the flagpole. It's quantitative. Whereas when you have categorical data, it's qualitative. All right. So another way to look at it, continuous data is usually measurements, whereas categorical data is categories. It makes sense. Another term for categorical is also discrete data, or yeah, that's the only one right now. There's a lot of terms, okay? And so just examples of what continuous data is: age, height, mass, length, temperature, concentrations of anything. And then for categorical, it would be sex, hair color, not just hair, morphologies, certain treatments that you set up for an experiment, or different sites. Those are things that are unchanged and you can't divide smaller. And we'll get into that later, because especially with continuous, you can really see it. Because for instance, the length of a fish, actually you can get down to 10 decimal places and it be slightly different from another one at that 10 decimal places. So it's pretty much infinitely, you can divide it up and it'll be different. So it's continuous and there's no specific break. And so at the bottom here, the type of data you have, so whether you have categorical or continuous data, determines what statistical test you will use. All right? And so in addition to that, when you have your data, you have two different types of variables. So your column usually is a variable. So in that spreadsheet, when you see a column, That's usually a variable. That's the term that is used, okay? And that's the type of data you collect. And so the terms that I prefer most are predictor variable and response variable, okay? And you'll see, because I think it makes the most sense. So you have the predictor variables, as you guys probably know, the independent variable, which you hear a lot. I remember hearing that when I was in grade school, and it never made sense to me. And then the dependent variable is the response variable. Again, that didn't make sense to me, so these terms were introduced to me in graduate school, and I like them a lot better. And so when you graph them, the predictor variable is the one that sits on the x-axis, and the response variable is the one that's on the y. So remember that for when you're doing graphs, because if you have the placement wrong, it won't make sense, because that's not what you're testing. All right? And so the reason it's called a predictor variable is because when you use a test, the predictor variable is the one that you're looking to see, does this affect that? So does fish weight affect the length? Okay, so which one is the one you're testing? So the response is the length, because you're asking, is this affecting this? The response is the second one, is the one being affected. All right? And so just like they say, it's dependent on the independent variable, but I hate the word independent, it makes no sense to me. And so similarly, my plant group does, how does plant biomass respond? So that's the response variable. How does it respond to nutrient levels? Okay, similar thing. So that's kind of the framework. And again, so statistics tests whether the relationship between these two variables is significant, and that's where it comes in. Because you have variables sometimes that are response variable. No, never mind. I was thinking back on the other slide. So your response variable is the one that you're trying to find out about. You're trying to find out what's going on here and what's causing it to be the way that it is, to weigh as much as it does, to be at whatever concentration it is, yada, 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 okay? All right, so making sense of data. There's two things that you'll see a lot that are really good statistical indicators or values that describe your data. And so the first one is the p-value, and that's the one that tells you if something's significant. So if you're looking at fish length and weight, is that relationship significant? Does fish length significantly predict the fish weight? Okay, so does it do a good job of telling you what the fish weight will be? Or the length, I don't remember this. Whatever. And so the rule of thumb is that if the p-value is less than 0.05, then your data is significant. Okay, and it doesn't matter if it's 0.04, it's still significant. If it's 0.000001, it's still significant. You don't want to use the terms highly significant or barely significant or almost significant or not at all significant. It's a contingency test, so it's not a spectrum. It's either it is or isn't. So you want to keep that in mind also. And there's actually a huge debate about that, but for the sake of what you're doing, that's the way it is. Okay? And then also, even though it might not be significant, you can still say, because you'll have means that might be similar and they're not, or they might be apart and different, but the error gets in the way, and so it's not significant, but it's still something that's different. You observe that the means are different. So it's still a conclusion that you can make. So don't get too hung up on significance. Okay? And then when you're doing regressions and correlations, R squared is a value that, and I should have done the superscript instead. It's used to describe the strength of the relationship. So how well does that line that you add to your regression, how well does that fit the data? All right, so that's an indicator of the strength of the significance. Because you can have something that's significant, and then the R squared is crap. Okay, and the closer it is to 1, the better. So it could be 0.01, and that means that your data doesn't really make sense. It might be significant, but it doesn't make sense. Yes?
Will it ever be above 1?
No. No, not at all.
Okay, or 0.
Or 0, yeah. It'll get really, really low. It'll get really, really close to 1, but there's essentially an asymptote there. So think about it that way. All right, yes?
Does that refer to a line of best fit, sort of?
Come again?
Yeah, that is.
Oh, the R squared,
Kind of
is that like a line of best fit?
Yeah, exactly. Yes. That's exactly what it is. So, all right. We good so far? You guys a little bored? Okay, well, I want you guys all to stand up. Okay. So just because it's dark, you know, it's the afternoon, you guys have lunch. I know they'd be excited about it, so I wanted to include them in the presentation. All right? So again, stay awake. You guys are doing great so far. All right. And so we have 4 main statistical tests that you guys will be using, okay? So you guys want to write this down because we'll go into examples of each using your hand data as well as your age and your grade. So we got that all. Thanks for putting that on the Google Doc. I was able to do all this last night and a little this morning. And so we have a regression, okay? What you have is you have continuous predictor variables. Okay, that's it. You don't have any discrete, and you can have as many as you want as long as you have at least one. Okay, because you can have a regression with 2 predictor variables, you can have one with 3, doesn't matter, as long as they're all continuous. Okay, so again, that's like height, mass, age, those types of things, temperature, nutrient concentrations, all right? And then a t-test is one you use with categorical data, but it's only when there's 2 categories. So things like yes or no. Did you enjoy the food? Yes or no. In order if you want to test that, you need to use the t-test. All right, so that's that test for that one. And again, it's predictor variables. Okay, the response variable doesn't matter if it is.
Is that part of the presentation?
It doesn't matter if it is a predictor. No, if it doesn't matter if it's continuous or discrete. The response doesn't matter. It's all about the predictor variables. Okay. Sorry, discrete is categorical. Oh my God. And then another one you could use is an ANOVA. Okay, so that's an analysis of variance. All right, so you guys are going to write that one down. Either analysis or variance, or just write ANOVA, and you can Google it, and you'll find the answer like in 5 seconds. Okay, same thing. And so that's basically it's a way to look at the variance in your data to then test significance as well. And so you have a predictor variable that is categorical, but it is composed of at least 3 categories. So it can't be yes or no. It can be yes, no, maybe. So if you're going to test like whether people enjoy $1 pizza day, you know, and you gave them options of yes, no, or maybe. Yeah, right. And so, although you guys were complaining because you guys missed it a little bit the other day, or whatever. Anyway, so then you do an ANOVA. You no longer do a t-test, instead you move to an ANOVA. Okay. And then the last one, which is the most complicated, and it's unlikely you guys will be doing this because not many people do, but just to throw it out there so that you can understand categorical data and continuous data coming together. And so for an ANCOVA, which is an analysis of covariance, okay, you use continuous data and categorical data to predict a response. Okay, so you'll have, let's say, yes or no, you like the pizza, and then the other one would be how many slices do you eat? Because that, you know, you could have an entire box. That's numbers and stuff. So, no, actually, you can have like half slices. Yeah, that's why it works. Okay. So you would, if you're using both of those to predict how full you were, or whether you come back again, if that's your response variable, whatever it is, you use an ANCOVA. All right. So keep that in mind. And so first things first. So this is the stuff that you had out on the sidewalk or whatever, the concrete. All right. So this is all your data: your hand span by your hand length. And so because the span is here, what does that mean? What variable is it? Predictor, yes. Okay, and then your response is on the y-axis. And so what test should we run for this type of thing? Look at our measurements, look at our units. Regression? Yes. Good job. And so you want to use a regression, and the way that, so this is the way that it's done on the computer with code, the way you write it. And the way I read it is hand length is predicted by hand span. Okay, so it's like a backwards equation. So if you want to write that down, that way you can kind of frame it. So hand length as predicted by hand span. That's just the framework, and that's how I'm going to continue writing all the other tests. Okay, and again, the tilde there is supposed to be just predicts. Okay, it's predicted by. Okay, and so when we do the regression, all right, there's the line, see the p-value, it's significant, so there is a significant relationship between the two, and the r-squared value is 0.3567, okay? It's not bad, you know, it's decent, especially when you get into biological data, set your bars lower, okay? Because nature never works the way it would in a laboratory. So this is just the way your hands are. A lot of people are sampling. That's a pretty good R-squared, in my opinion, for that type of experiment that was done quickly, probably not all that well, and with different standards for people, okay? So science is messy. It's okay. Listen to Miss Frizzle. All right? Okay. And so now this is when you guys kind of like break into your group and you guys are asking, okay, are you guys going to use a regression? And if so, why and how? So think about your variables that you collected while you guys are out in the field. Are any of them both continuous? So think about that. All right? So
Discuss for about 2 minutes.
yeah.
Yeah?
Okay.
Awesome.
Good. And keep in mind, you might use multiple of these, okay? You might use all of them, one of them, two of them, doesn't matter. All right? So how can you test hand length and span at the same time if you're trying to look at the effect of sex? So if you're trying to look at your entire, like instead of just, if you're trying to incorporate both variables into it, instead of just looking at length or just span, trying to see if sex predicts your hand size, what can you do? Yes? No, not a test. Like, so what can you do to the data?
Color coded.
No. Yes?
Categorize?
No. Okay, so you can transform it. You can change it and make it so that you incorporate them. The easiest way to do that is to create a new variable. And so if you multiply hand length by hand span, you get the hand area. And so both of those are part of hand area. So you're testing both of them at the same time because they're both parts of area. Okay? So do you understand? Like the size of your hands, you're getting everything. So all of a sudden your units change because now you're working with an area, you have centimeters squared. And so you're testing both of them. So this is the best way to do it. Otherwise you could test them independently. Test, hand span and see if it's predicted well by gender, or you could do hand length separately, but the best way to incorporate them both at the same time, kill two birds with one stone, is to multiply them. Alright? So, sorry I used typical gender colors. Okay, I heard the chalk argument earlier. So this is what we have. This is what it comes out to. Alright? And so, what tests are we running here? Louder? Yeah, t-test. Good. Okay, because you only have two categories. And it's categorical. Alright? And so the way it is, so you do a t-test with your hand area as predicted by your sex. And that's what you're testing here. Okay? And so you see the error bars, those are for standard error. And so that incorporates your standard deviation, which is basically just the spread of your data. How far apart is it? And so you're going to have larger standard error in the guys, because there's only 5 of them, whereas with the girls there were 17. Okay? So usually as you get higher numbers, you get tighter standard error. It's smaller. Your standard deviation is smaller, your variance is smaller. When you have less, you have greater variance in everything. Okay? That's why it's always better to get as much data as possible, as many, your N, you want it to be as huge as possible. Makes it a lot better. Alright? And so for this test, all you get out is the p-value. There's other things, all you really care about is your p-value. And you see it's significantly different. Okay, visually a good way to tell is if the error bars overlap. Usually it isn't. But you still need to run the test to double check. Alright, because you don't really know, because there might be infinitely small and it's still really different. But here, you can tell, yes, they're different and significantly. Okay, so now think, are you guys using a t-test? Why? How? Talk amongst yourselves. Okay? Proceed. Alrighty. And then so next suggestion. Alright, and so what test would we be running here? Yes, an ANOVA. Okay, and why? Yes, because there's more than 2 categories, okay? You can have 17 categories. Doesn't matter. It's more than 2. All right? And so what we see here, we run an ANOVA with hand area as predicted by your grade. Okay? And so what we see is a p-value of 0.687, which means they do not significantly differ. All right? Now, after you do an ANOVA, if you want to test the relationship between these two as individuals, what you do is called a pairwise t-test. Okay? I ran one really quickly on this, and none of them were significant. But sometimes you might have something like, let's say it starts, this one's like really high, this one's in the middle, and this one's below. and the entire thing is insignificant, but you want to test the two ends, then you use your pairwise t-test for that. All right, it's a post hoc test. It's something you do after you run the initial test to get more information. All right, so that's an ANOVA, pretty simple. You can also have what are called two-way ANOVAs. So you have two variables that are discrete or categorical, trying to get the same, trying to see if they predict your hand area. So let's say if we did hair color and grade. Okay, those, I mean, I guess hair color can be kind of continuous, but it's not numerically, so it doesn't matter. And so if you use both of those to try to predict your hand area, then you would use what's called a two-way ANOVA. So this is a one-way ANOVA. And you can have different levels. Okay, so just keep that in mind, but that's how an ANOVA works, and that's what you're looking at. All right, so now talk amongst your group. Are you guys doing an ANOVA or not? If you are, why? And how are you going to use it? What variables are you going to use? Kind of make that sense. And don't hesitate to ask questions, like I predicts hand area. Okay? This is the data. What test do we run? What? Okay, so you know what that tells me when I hear ANCOVA? Is that you're not thinking about the problem, you're just looking at the next one on the list. Alright? So, it's not an ANCOVA. Sam, what did you say it was? Yes, it's a regression. Okay, Sam, why is it a regression? Yes, they're both, it's not just that they're numbers, they're both continuous. Okay? So even though these ages only fall into certain ranges, so they're only 16, 17, 18, 15, that's because that's what we knew. But someone could be 17 in a day, or 17 and a half. And so it's continuous. You can slice it up. You can cut it up and make it smaller. Okay, and that's kind of what makes it continuous, and then your response is also continuous. Alright? So again, as a reminder, an ANCOVA is with categorical and continuous. There is no categorical here. It's only continuous, so we just use a regression. So I was trying to trick you guys. Alright? And I did. So regression, okay, so you're looking at hand area and whether it's predicted by age. Okay, again, similar way to write it. Okay. And then what you have here is you have a p-value of 0.862. So it is not significant. Okay, and the r-squared value is really low. So that's saying that this line very poorly describes the data. Okay? And so there really is no relationship between any of these things. Okay, that's what this all says. So p-value not significant, and a bad r-squared, there's absolutely no relationship at all. Okay? And it's totally okay to not have significant results. This is something that even graduate students that graduated before me that I saw in our lab panicking, they're like, oh yes, sorry.
No, it's okay. I was just wondering, so when you said the r-squared was lower in showing the line slope was pretty low. Does that have an effect on the r-squared?
Slope doesn't necessarily have an effect.
No.
So like if all those lines, if all those points were clustered closely here, then it would have a really strong r-squared.
Yeah, okay.
So the fact that they're so spaced out, because the line is calculated by taking the distance from the point to the line and factoring that all in. And so they're far apart and not that related, so that's why you have a really bad r-squared. Okay?
I see.
And so like there was a graduate student that graduated last semester or the semester before, and she kept on panicking because her results weren't significant. She didn't have a p-value of less than 0.05, but I want you guys now at your age and everything to get into your head. Just significance, statistical significance isn't what's important. It's your conclusion that you draw from that. Okay, so if you have a result that is not statistically significant, you're still finding out, okay, so nutrient levels do not predict plant biomass. You now know that there is no correlation there, and so you know what you didn't know before, that those nutrient values do not predict biomass. Okay? Whereas if it were significant, you just found out, okay, I didn't know anything about the relationship between these two, and now I know that they are related. Otherwise, you find out if it's not significant, they're not related. So it's still an answer to your question. And that's the most important thing. That's what statistics does. It answers your questions that you ask. If you're doing it with respect to either, even if it's descriptive, you're finding out about those things and those variables. Okay? Okay, so if we're looking at two predictor variables now, okay, what do we have? Predictor variables, not categories. Okay, So the answer is it could be either an ANCOVA, a two-way ANOVA, or another regression. Okay? It depends what type. So let's see what we have in this example. All right, so what do you see on this graph that kind of can tell things and create whatever? So what do you see? You have male and female, so you see a category. What else do you see? Two continuous variables, okay? So what do we do with those, with that setup? When we have categories and continuous, what do we have? An ANCOVA, exactly. Okay, and so the way this is written out is hand span is predicted by hand length and sex. So you're incorporating both of those into the test. All right? And the way you do this is just look at it like this. So you draw both lines for the regression, and then what you're doing is you're comparing the lines. Essentially, that's the simple way to think about it. And so what we see here is we have a p-value of 0.89435. Okay? And that's the p-value for the interaction between the two terms working together to predict it. Okay, so sex and what was the other one? Hand span do not predict your hand length. However, if you were to separate it out, maybe your sex does predict it independently of the hand span, but together they don't. Okay? Yes?
Why don't they? Oh, never mind.
Yeah, it's above 0.05. Yeah, it's not an R squared, it's p-value. Okay? And so the R squared here, if you're going to do an R squared, you would basically be running two regressions, a regression for just the females and a regression for just the males. So I did that and I decided to take it out just because it was getting really messy on the PowerPoint. But there's a lot you can do with these things and test them that way. All right? And so what you guys to think about, are you guys using an ANCOVA at all? Okay? And then this is also when you want to ask, are you doing a two-way ANOVA? Are you doing a regression with multiple factors? Okay, so how many variables are you incorporating into your test that you're doing? All right? This is what you want to write down. Okay, remember, two categories only. And you want to remember minimum, minimum, minimum. And for ANOVAs, you're going to have more than one predictor variable. And you also have more than one predictor variable for a regression. For an ANCOVA, you need more than one, and they need to be different. All right? So keep those things in mind. Your mentors will be really impressed if you tell them, we need to do a two-way ANOVA. Okay? They're just going to be shocked because they're like, whoa, what do you mean? And then you say, well, we have gender or sex as one variable, and then the other one that we have is where you live, what city, and you're trying to predict your shoe size. Okay, like I really don't know. And so, but that's something because you have two categorical variables. You're trying to see if they're at all related to a that's a predictive variable, right? Yeah, two categorical predictive variables trying to predict a response that's continuous. Okay, but you can also use continuous variables to predict a response that is, I was going to say, discrete, categorical. So you can switch between those things. And it doesn't matter what the response is. It doesn't matter what type the response is. What matters is what type the predictors are. All right? So any questions at all about your specific case? Anything you want to ask me?